Hi everyone, thanks for joining us. I'm Shannon Barrett, Communications Officer here at WeedSmart, and today I'm joined by Dr Catherine Borger. Catherine is a research scientist from Western Australia's Department of Primary Industries and Regional Development, otherwise known as DPIRD, and Catherine's research has a particular focus on weeds. So before I hand over to Catherine to talk about her recent work with brome and barley grass, I'd just like to introduce WeedSmart for those unfamiliar. So WeedSmart's an industry-funded communications platform delivering science-backed weed control solutions. And on the screen, you'll be able to see our partners, and all of those above the line are our financial partners, and everybody below the line are our in-kind partners. So a huge thank you to the contribution from these companies. Catherine, I'll hand over to you now, and if any attendees have any questions along the way, please utilise the Q&A box at the bottom of the screen. Over to you, Catherine.
Thank you. Let me just get this talk up. Okay, welcome everyone. Thanks for, I can't say coming along today, but tuning in. This talk is about grape brome and barley grass, trying to understand the enemy. Oh, my clicker stopped working. There we go, all fixed. Okay. Why are grape brome and barley grass bad? Grape brome, in the last 10 years or so, has snuck up to our fourth most important weed nationally. It costs us about $22 million a year nationally. Barley grass is not quite as bad as that. Barley grass is pretty reliably in our list of top 10 worst weeds. It probably costs us about $2 million a year annually. But it's still certainly a problem over some very large areas. Why are we researching them? Well, because you told us to. This is a national project funded by the GRDC and directed by Gurjit Gill at University of Adelaide. It also has collaborators at University of Queensland and in New South Wales. All of these groups were given lists of weeds to study, and the lists were devised by the RCSN groups. So these are all weed species that growers felt they needed more information on ecology and biology before they could formulate effective integrated weed management programs. So the research areas we focused on included how long does a seed bank last? When do these weed species emerge? What are they doing to the crop? When do they shed seed? And in the case of brome and barley grass, is harvest weed seed control any good at all for species that might shed all their seed? First question, how long does a seed bank last? Well, we tested this in a few different ways. We got 12 different populations of brome and barley grass and put them in irrigated conditions. So perfect germination conditions, the seeds are at the right depth, they stayed nice and wet. And our emergence of both species as a percent of the total available seed was around 40% in the first year and 6 to 12% in the second year and very, very low by the third year. So in perfect conditions, assuming nothing goes wrong, about 2 years of really good control is enough to control these weed species. We did a similar test out at the field at Wongan Hills. We planted a few populations of barley grass and grape brome, 1,000 seeds per metre squared for each population. And we found that on average, out of those 1,000 seeds per metre, we had about 400 barley grass seeds come up, or 40% of the population, and about 66% of the grape brome come up in the first year, 2016. In the second year, we had 3 to 7% of the weeds come up, and there was still a reasonable amount of emergence in the third year, and a few struggling weeds coming up in the fourth year. It's much easier for weeds not to germinate in the field. The process of sowing the crop is going to bury some of the weed seeds. They will be too deep to germinate. Burying weed seeds tends to induce dormancy, so they can sit there for longer. Some of the seeds are protected by crop residue or in little pockets of non-wetting soil, so they're not getting enough moisture. So in these field conditions, we really saw that you need at least 3 years of control and 4 years for some populations to really get your barley grass and grape brome under control. Next is, when do they emerge? Here's some barley grass populations that we collected, 6 different populations. And for each population, we took some seed from the roadside and we took a matching population of seed from the crop next to the roadside, sort of 20 meters away from the fence line or roadside. Then we planted them at the end of April. And this shows cumulative emergence of those barley grass populations that were collected from the roadside. Essentially, they went in the ground, they got wet, and they germinated. Most populations had over 90% germination in the first week they were in the ground. A couple of populations only had 70 or 80% emergence in the first week, but emergence was really high and fairly quick. Here are the same populations we collected from the roads, from the crop instead of the roadside. And you can see that some of the crop populations were responding in the same way as the roadside populations. As soon as they went in the soil and got wet, they germinated. But some of our crop populations had significantly delayed germination. Some of the populations kept having cohorts right through May, June, July, August, and even September of that year. So this looks like a deliberate strategy the weeds have used to avoid herbicides. Obviously, seedlings germinating in June and July and August are not going to get hit by knockdown herbicides, pre-seeding herbicides, and even the in-crop herbicides are going to be struggling to target all of those different cohorts. So it is a great way to avoid herbicide, but it's a very, very expensive strategy for a weed to adopt. A plant germinating in July or August is going to be a very small weed in a fairly big mature crop. So it's going to be very poorly competitive. Seed production will be reduced, but those late cohorts can still set some seed and keep going next year. I'm not going to show you this same graph for grape brome because all the grape brome populations pretty much looked like barley grass populations from the roadside. We planted the seed, they got wet and cold in April, they immediately germinated. So in grape brome, we weren't regularly seeing the same delayed emergence, except in one population. By comparison, when they did a similar experiment in South Australia, they found their grape brome from the field pretty reliably had delayed emergence. So possibly the South Australian grape brome populations have just been managed intensively for a longer period than the grape brome in WA. And it's possible that our WA populations in future will evolve the same delayed emergence strategy, or they might just keep emerging early and evolve herbicide resistance instead. You never really know with weeds. The next question was, what do these weeds do to the crop? Well, this shows a wheat crop grown in Wongan Hills in 2019. Mace wheat, it wasn't a fantastic year. You can see we've got weed densities at 50 to 200 barley grass plants, 50 to 150 grape brome plants. Even where the weed density was 0, the wheat yield was only about 1.5 tonnes. It wasn't a very good year in 2019. You can see as weed density increases, barley grass reduced yield by a bit over 30%, grape brome reduced yield by a bit over 40%, which is really not what you want, especially in a dry year. By comparison, let's look at what happened in 2017. This is the same maize wheat in Wongan Hills. Not exactly the same site, but it was the same field, same soil type. In 2017, it was a better year. You can see we had over 200 barley and grape brome grass seed plants of a high density. But you can see where wheat density was 0, the wheat yield was closer to 2 tonnes per hectare than 1.5 tonnes. So high yielding crop. The other thing that happened in 2017 was that Wongan Hills got a little bit of summer rainfall, so there was moisture at depth, but then there was a very, very dry autumn. So the crop was dry sown into moisture. The crop came up pretty easily. The weed seeds kept sitting there on the soil surface for 3 weeks. At one stage, we thought the crop was going to die because it took a long time to get a bit more rainfall. But we got the rainfall, the crop hung in there, the weeds came up pretty late, and even 200, 250 plants per metre squared had no impact on wheat yield. Most years, weeds will affect the yield. But for a different project a year ago, I surveyed growers about barley grass, and I found that there was a perception among a lot of people that barley grass didn't really affect crop yield. And that's because they've all seen the years like 2017, where barley grass does not affect your crop yield. A lot of barley grass in WA can be quite short, quite uncompetitive. It's a lot like annual ryegrass. We have a lot of trials to show that ryegrass is competitive in crop and reduces yield. But there's quite a lot of other trials that show that ryegrass is not that competitive and doesn't always have a big impact on yield because they're quite short, spindly plants. So I put these 2 graphs up there to highlight that, yes, there are plenty of years where barley grass in particular might not have a huge impact on crop yield, but we control the weeds for those years like 2019. They're a bit drier and the crop's struggling a bit, and you really don't need weeds impacting your yield. Next question, when do they shed seed? We looked at seasonal variation of grape brome and barley grass shedding. Again, this was a trial at Wongan Hills. We grew grape brome in a wheat crop. And then we looked at the percent of seed retained from about October to harvest. So you can see I've got 3 different years on this graph, 2016, 2017, and 2018. For each year, the line stops when we harvested the crop. So for 2016, the line stops on the 1st of December when we harvested. And by the 1st of December in 2016, all of the grape brome seed had shed. There wasn't a single seed left on a plant anywhere in that field. By comparison, in 2017 and 2018, there was still some seed on the plants at harvest, but that was mainly because harvest was a bit earlier in those 2 years. You can see we were harvesting end of November, not start of December. It's worth noting that in all 3 years, we could have harvested around mid-November. That was the earliest possible harvest date for crop maturity. And you can see by mid-November, there was 40 to 70% seed retained. So not high seed retention, but if you can harvest your brome grass fields early as a priority, then you can probably capture some of the seed.
Catherine, I've just had a question come through.
Sure.
In 2019, did the weeds come up with the crop versus later in 2017?
No, in all 3 years, our grape brome came up with the crop. It was highly competitive. They emerged at the same time, and I'd go out a week after emergence and I'd have tiny little mace wheat, maybe 5 centimetres high, and the first grape brome cohort was already at 10 or 15 centimetres, towering over the crop, but always very early, vigorous emergence.
Thank you.
So this slight difference is just the seasonal differences that we saw later on in each year. Barley grass had a lot of variation. It was a bit mysterious. This is the same graph as for the grape brome. And you can see that in 2016, barley grass by harvest date on 1st of December had 100% of seed shed. There was not a single seed left on a plant anywhere. In 2017, there was over 90% of the seeds left on the plant. And in 2018, we had 100% seed retention on the plants. To be honest, I was a bit confused. I was standing out there in the field, giving the seed heads of the barley grass a little poke. Then I gave them a little tug. Then I kicked them a bit and hit them with a shovel, but those weed seeds were not falling off. And for the 2018 crop, I actually came back the following February. And I took a picture of the brome grass. You can see the picture here. It's a little bit unclear because what we have is dead weeds against dead wheat stubble. But if you look closely, you can see that by February, all the seeds were still sitting there on their seed heads, firmly attached. This really shows that there's a lot we don't know about how seed shedding works. The traditional idea around seed shedding is that you get hot weather with low humidity, and then you have a bit of wind and the seeds fall off once they're dead. Which is why seed shedding usually occurs around mid-afternoon. You have highest temperature, lowest humidity, and unless you're having a storm or unusual event, wind speed is generally greatest around mid-afternoon. For barley grass, obviously you need some hot, dry weather to get senescence, but obviously there's a combination of seasonal conditions that occur when the seeds are forming, and if you don't get the right climatic conditions at the right time, the seed stays put. Because I didn't clarify at the start, but these 3 years were using the same populations of weeds. There was no genetic variation. This is just a seasonal variation. So we clearly need a bit more research in why weed seeds shed when they do. This graph looks at the genetic variation in seed shedding. For this particular trial, we collected barley grass from several different populations at several different sites around the WA wheat belt. You can see we've got a few different populations from Merredin, a few around Northern York Grass Valley, a few out towards Wongan Hills. We collected the seeds in 2017 from these different populations, and then we grew them up in controlled conditions in 2018, and we monitored their shedding. Now, I've called this graph genetic variation, but it would be more accurate to say it's genetic and environmental variation. We might have seen a difference in shedding due to genetic differences between these populations. We might also be seeing this difference in shedding because the parent plants that we collected the seed from in 2017 experienced different environments, and the environment experienced by the parent plant can then affect what the seed does in the following season. So let's call it combined genetic and environmental variation. But that's still worth looking at because the combined genetic and environmental variation is what the growers have to deal with. So this is showing maximum variation that we're seeing in barley grass shedding. So in 2018, the seed started shedding around October. And we saw that in this particular year, all the seed had shed by mid-December, which was similar to what we saw in the field in 2016 at Wongan Hills. You can see some populations just shed seed steadily from October to December. Some populations didn't actually start shedding seed until November, but then they shed fairly quickly as it heated up. So the main finding here is there's quite a lot of variation in the shedding time of barley grass. I could show you a similar graph for grape brome, but it pretty much looks the same. Slightly less variation, but similar trends.
We've had another question come through, Catherine, just on your last slide. Were there any populations from southern WA?
No, we didn't get down to southern WA. but I'm sure that would have a different pattern again. Then we looked at harvest weed seed control, because we've just established that these two populations have very variable shedding, but the bottom line was that in some years they could shed a lot of seed. There wasn't much left on the plant to harvest. So does harvest weed seed control have any value here at all? We used all the weed ecology data we had collected over the last 5 years. Then we took the weed ecology data that South Australia had collected over the same 5-year period, and we used it to update the Weed Seed Wizard model. And if you want all the fine details of how we changed the model, it's in a Grains Research Updates paper from this year. We modelled a rotation of wheat, lentils over 6 years at Wongan Hills. I'd just like to highlight here that this is hypothetical, particularly for barley grass. We have not run any field tests to show how well barley grass actually goes into the harvester. Even brome grass, some growers are using harvest weed seed control for great brome grass, and there's really variable results. For example, Nick Fells down in Esperance finds that harvest weed seed control is pretty good for his great brome. Kit Leek in Calabaron found that it wasn't particularly good for his great brome. The plants were still green at harvest. He would try to harvest them, but because the plants were still green, they just bend forward and slip under the harvester. I tried suggesting he could harvest a bit slower in that patch, and he told me that was a dumb idea. No, that's not true. He didn't say dumb. Kit's a gentleman. He probably just meant dumb. He definitely established that he wouldn't be harvesting a bit slower. So our rotation, as I said, was 6 years. It was a rotation with normal herbicides for the Wongan Hills area. In 2011, we, in the model, sprayed some glyphosate, then some spray seed and treflan, sowed some wheat, put some Midas on the wheat, harvested the wheat. In 2012, we sprayed some glyphosate, then some trifluralin and Monza. Then again, we sowed and harvested wheat. In 2013, we had a double knock, glyphosate, then spray seed. Then we applied some Bladex. Then we sowed the lentils, put clethodon on lentils in the crop, and harvested. And in 2014, 2015, and 2016, we did exactly the same things. So again, wheat, lentils. This graph shows our results of harvest weed seed control for grape brome. So in the model, we started off with 100 seeds per metre squared in the soil seed bank. Over the 6-year rotation, we either had 1% harvest weed seed control, which is your normal harvest. In a normal harvest with all chaff spread out to the back, you're going to have 1% of your weed seed captured in the grain sample as a contaminant. Then we tried harvest weed seed control, assuming we only caught 20% of the grape brome grass seeds, 40% up to 100% of grape brome seed destroyed. So you can see where we had 1% seed destroyed. We started, as I said, with 100 seeds per metre squared soil seed bank. And by the end of 2011, the first year of our wheat, we were up to 300 seeds in the soil seed bank. And by the end of our 6-year rotation, we had almost 11,000 seeds in the soil seed bank. So the grape brome grass really got away from us there. This was clearly a bad rotation, and if we were doing it in real life, we would seriously think about our agronomic package. But anyway, it doesn't really matter what the agronomic package or the rotation was. What really matters here is that if we assumed that 20% of that seed got caught and destroyed at harvest, then we only had 6,000 grape brome seeds in the soil seed bank at the end of our 6-year rotation. And from a practical point of view, 6,000 seeds in your seed bank is still pretty bad, but it's almost half as many seeds as we had when we only had 1% of the seed destroyed at harvest. And if we go up to 60% harvest weed seed control, we actually had our seed bank at the end of our 6-year rotation down to 86 seeds. So that was lower than the number of seeds we started with. We're making progress. As I said, for this data, it doesn't really matter what the rotation was. And you can't take away from this data that 60% seed destruction is a magical number for grape brome, and as long as you achieve that, your population will go down. What this data is showing is how much difference you can get to your final seed bank from even a tiny percent of weed seed control. We all know at the start of the year that controlling 20% of the weed plants makes no difference. If there was a herbicide that only controlled 20% of seedlings, you would not buy it. You know that if you kill 20% of the seeds at the start, the population just recovers, the surviving plants grow bigger, they produce more seed, and you make absolutely no difference. But destroying the weed seeds at the end of the year is a lot more powerful. The population cannot recover from that. So even 20 or 40% weed seed control at harvest can still make a big difference to your long-term seed bank. And in most years, we saw that by crop maturity, grape brome had 20 to 70% seed retained. So there is an opportunity there to kill some of that seed. We ran the same rotation for our barley grass, and you can see that if we started off with 100 barley grass seeds in the soil seed bank at the start of the rotation, with normal harvest, we only had 28 seeds by 2011. And by 2016, the population was effectively eradicated. So harvest weed seed control was still good at controlling barley grass. Obviously, 20, 40, 60% of seed destruction at harvest helped us run the seed bank down a lot faster than no harvest weed seed control. But essentially, barley grass in WA is a much less competitive seedling than grape brome. The plants tend to be shorter and less competitive in the crop. So with this particular rotation with normal herbicide use, barley grass was not really a problem. In conclusion, from this research, we found that the soil seed bank lasts 3 to 4 years. It depends a bit on population, so you really need to know what your own population is doing. Both these weeds, but particularly barley grass, could have very staggered emergence, which makes it really difficult to kill all the weeds at the start of the year. It does mean that any competitive late weed cohorts are going to be less competitive with the crop. But if you want to stop seed set, you still need some kind of plan very late in the season to get the weed seeds of those survivors. The competitive ability of these weed species varied pretty widely between years. But in 2019, economic returns from the Mace wheat at Wongman Hills were reduced by $90 to $205 per hectare by grape brome and $67 to $138 per hectare by barley grass. So that's quite a lot of lost profit. The time of seed shedding is really variable. So harvest weed seed control is still effective, even if you can only catch a small percent of the weed seed. But you really need to go out and have a look at what your own population's doing each year to get a feel for how effective harvest weed seed control will be. And we really do need a lot more field research to find out how effective harvest weed seed control is in real life. Are those seed heads actually going into the harvester? Or are they lodging? Are they too green? Are they just too short to get in the harvester easily? Thank you for attending this talk. And I would like to thank all the staff who have helped with this research over the last 5 years. 5 years is quite a long time in research years. This project actually started with Abul Hashem. I was on maternity leave, I wasn't here at all. Abul has now retired, so I get to finish off this project. And Glenn Reithmuller was also present at the start of this project and is unfortunately absent for the end of the project. I'd like to thank Gurjit Gill and Daniel Peterson at the University of Adelaide, and a range of other people, particularly Shari Dougal and Bruce Thorpe at the Wonga Hills Research Station, because I did spend the last 5 years planting a lot of different weeds on their research station, which they accepted really gracefully. Thank you. Any questions?
Awesome. Thanks so much, Catherine. We have had a couple of questions come through. We actually had a question come through from one of your colleagues about a month ago. So I've had that one on the back burner. So what is the status of glyphosate resistance in brome grass and any other resistance statuses if relevant?
Okay, in Great Brome, we do have glyphosate resistance in the eastern states. We also have resistance to ACC herbicides, the group A, hops and dims, and some resistance to the ALS herbicides like metsulfuron methyl. In barley grass, again, we have resistance to the group A herbicides. Barley grass, Hordeum leporum has shown some resistance to paraquat diquat spray seed, and Hordeum glaucum species has also shown some resistance to glyphosate. These resistant populations are most common in the eastern states. South Australia in particular has big resistance problems for both these weeds. WA has lower levels of resistance. Michelle Owen, in her last RE survey of weeds, found some low levels of resistance. I checked resistance in barley grass last year and didn't find any, but we'll check again this year. So resistance levels are lower than weeds like annual ryegrass and wild radish, but they're certainly increasing fairly rapidly.
Yeah, great, thanks. I think Michelle's data has actually been updated on the RE website as well, if anybody wanted to check that out. And we've had another question come through. How applicable are the WA-based findings to the eastern states?
Well, I think for Great Brome, there's a lot of similarities. As I mentioned, some of the South Australian Great Brome populations have now developed delayed emergence, which our populations aren't seeing as commonly. But Great Brome in both WA and South Australia, they're very competitive against wheat. They do have earlier emergence. They both have very, very high seed set if they're not controlled by chemicals. Barley grass is a lot more variable. We don't have one species of barley grass. As I mentioned in that last question, we have Hordeum leporum and Hordeum glaucum. In South Australia, Hordeum glaucum is by far the most common species. In WA, we have both species distributed about equally, and it's common to find both species in the same field, in the same sward of grass. You can't really easily tell the difference between them. You need taxonomic skills and a microscope. And frankly, there's now some research to suggest that the morphological markers we use to tell the two species apart are not consistently different between those two species. And you can have a look at their chromosome number, but that can also be variable. Because it's so hard to tell the species apart, herbicide labels tend to just have a generic use pattern for barley grass without specifying a species. But the species themselves have quite a lot of difference. For example, our barley grass populations, I grew both species in various trials that I showed you in this talk. And our populations tended to range between about 12 and 20 to 30 centimetres high. And by comparison, the barley grass in South Australia was pretty commonly at the same height as the crop canopy and tended to be more competitive with higher seed production. So the variation we've seen between populations, between the different states and within the states, really show that farmers need to be their own weed ecologist. It was much easier with ryegrass. We found something out about the ryegrass. We went to tell farmers. We gave ourselves a little pat on the back at the end of the research. Job well done. We can move on to the next thing. But the amount of variation we've seen in these weed species really highlights that farmers just have to be out there in the field to look at what their own weed species are doing.
Okay, great. We've had a couple of more questions come through. Sorry, I'm not going to pronounce these correctly, but is there a difference between Bromus diandrus and Bromus rigidus?
Well, that's an international debate. I've just drafted a fascinating review on brome grass. In Australia, we tend to think of them as separate species because we have less genetic diversity than the rest of the world. We find that Bromus rigidus has a tighter, more compact head, and Bromus diandrus has an open, nodding head, more like an oak plant. And we have different and consistent chromosome numbers between the two. But in other parts of the world, there's different ploidy levels, which means there's different numbers of chromosomes within the same species. And they also find that that head shape, compact versus open and nodding, is not consistently associated with a certain chromosome number or a certain population type. So in the US, they tend to consider them the same species. Well, they consider them two different subspecies within a broader species that also includes Bromus sterilis. In Europe, parts of Europe assume they're the same species. In the UK, they're called by a different species and genus name, and they are treated as separate species generally. But honestly, we need a lot more research into the genetics of these species on an international level before we can find out if they're one species or two species. And even in WA, we see some evidence of crossbreeding and some mixed populations. So it might not be as simple as, are they one species or two? They might be in the process of speciation, which means they're turning into two species or turning back into one species. So to sum up, I don't know. Nobody knows.
Awesome. Thanks so much, Catherine. And thanks for joining us today and everyone's great questions.